In this video, we're going to cover drug-receptor interactions. We'll explore important concepts like drug binding, affinity and efficacy, concentration response curves, and we'll also break down the role of agonist drugs and dive into the different types of antagonism. Let's start this lecture by understanding the concept of drug-receptor interaction. If you've seen the previous lecture, we covered how receptors are specialized proteins located on cell surfaces or within cells that are responsible for transmitting signals in the body. When a drug interacts with a receptor, it can either activate or inhibit the receptor's function, leading to various physiological responses. There are four main types of receptor superfamilies: ligand-gated ion channels, GPCRs, kinase-linked receptors, and nuclear receptors. Think of receptors as tiny locks on the surface of cells or within cells. When a ligand, such as a medication or a naturally occurring substance, such as a neurotransmitter or a hormone, comes into contact with the receptor, it fits into the receptor's lock like a key. This interaction between the ligand and the receptor is called binding. Essentially, all your different types of receptor proteins will have a particular 3D structure, and they have multiple molecular domains. When that comes together in 3 dimensions, it creates some sort of pocket or binding site that will allow complementary ligands to bind. We refer to these structures that are created as the receptor binding sites or the ligand binding sites. So when the ligand binds to the receptor, it triggers a series of events within a cell. These events can include activating certain genes, altering enzyme activity, or regulating the release of other chemical messengers. The overall outcome depends on the type of receptor and the specific ligand that binds to it. In most cases, for a drug to have an effect, it needs to interact with a particular molecular target. With that said, what influences these drug receptor binding sites? Number one is the chemical structure. Okay, so the chemical structure of a drug determines its ability to interact with the receptor. It's going to be influenced by the 3D structure of the receptor, which will be influenced by the protein sequence. Whether or not the different amino acid residues are hydrophilic or hydrophobic, the different intramolecular forces will all influence that binding site. Okay. Secondly, molecular size and shape. The size and shape of a drug molecule influences its ability to fit into the receptor's binding site. Drugs that closely match the shape and size of the receptor's binding site have a higher probability of binding. We can also add the lipid solubility of a drug affects its ability to cross cell membranes and reach the target receptor. Lipophilic drugs have an easier time passing through lipid-rich cellular membranes, okay, improving their access to receptors. And then pH and ionization. So the ionization state of a drug is influenced by the pH of its environment. Ionized drugs have different binding properties than their non-ionized counterparts, so they may interact with receptors differently based on their charge. All right? Now, when we talk about drugs, we often need them to interact with specific targets in the body to produce their effects. However, it's important to understand that just because a drug binds to a receptor doesn't automatically mean that the receptor will be activated. There are 2 separate steps involved: drug binding and receptor activation. So now let's subtract complexity and go through receptor binding. The tendency of a drug to bind to a receptor is determined by its affinity for that receptor. Okay? So affinity refers to the tendency of a drug to bind to its receptor and the strength of the interactions. We mentioned earlier that factors such as structural compatibility, size, shape, charge, and 3-dimensional structure of the protein influence whether a drug can bind to the receptor. The stronger the interaction between the drug and its binding site on the receptor, the higher the affinity, okay? So a drug with high affinity has a greater tendency to bind to the receptor and will do so easily. Whereas a drug with low affinity won't bind to the receptor as effectively. So the better the affinity, the more drug molecules will be bound to the receptor. But the question is, how do we measure this? So to measure the affinity between a drug and its receptor, we use a parameter known as the equilibrium dissociation constant, KD. So this value quantifies the affinity by representing the concentration of drug at which half of the receptors are occupy. Okay, so in simpler terms, a lower KD indicates a higher affinity between the drug and the receptor. So understanding the affinity of a drug for its receptor is crucial in pharmacology because it helps us predict how strongly a drug will bind to its target and how effective it will be in producing the desired therapeutic effects. So before we break down the dissociation constant, let's first understand what drives the binding of drugs to receptors. This is really important. This binding process follows a principle called the law of mass action, which you may have come across before, okay? The law of mass action states that the rate of a reaction is proportional to the concentrations of the reactants involved. So, in other words, changes in the concentration of any component in the reaction will affect the rate at which the reaction progresses and where equilibrium is reached. To illustrate this, let's consider the interaction between a drug, okay, D, and a receptor, R. When they bind together, they form a drug-receptor complex, DR, this right here. On one side, you have the drug and receptor separate and unbound, okay? And on the other side, you have the drug-receptor complex. If the concentration of the drug is increased by adding more of it to the system, the higher concentration of the drug drives the reaction towards the formation of more drug-receptor complexes. So essentially, more drug molecules bind to receptors, resulting in the formation of more drug-receptor complexes, okay? And changes in the number of receptors can influence the reaction. However, in the field of pharmacology, we typically focus on changes in the concentration of the drug rather than changes in the concentration of the receptor, as receptor numbers usually remain relatively constant, okay, in biological systems. Now, another important aspect of drug-receptor interactions is that the formation of the drug-receptor complex is reversible. This means that while the drug can bind to the receptor and form the complex right here, it can also dissociate from the receptor, separating into the individual drug and receptor again. This reversible nature applies to most drug-receptor interactions, although there are some exceptions that we'll discuss later. Now, in most cases, the binding of drugs to receptor is reversible reaction, meaning it can occur through association or dissociation. The rate constants, K1 and K-1, describes the rate at which the drug and receptor associate or bind together and dissociate, separate. Okay, so K1 represents the association rate constant. So it determines the ability of the drug and receptor to bind, while K1 represents the dissociation rate constant, describing the rate at which the drug-receptor complex separates. All right? As you may have studied before, when the reaction reaches equilibrium, the forward and backward rates are equal. At this point, the rate of drug-receptor association is the same as the rate of dissociation. The equilibrium dissociation constant, KD, is a measure of affinity. It's calculated as the ratio of the dissociation constant, K-1, to the association constant, K1. So KD indicates the tendency of a drug to dissociate or separate from its receptor. Now, the value of KD increases when the dissociation constant, K-1, is higher. And the units of KD are in molar, which are the same units used for concentration. Numerically, the KD, or equilibrium dissociation constant, represents the amount of drug needed to occupy 50% of the receptor binding sites at equilibrium, okay? So this value provides us with valuable information about drug affinity. Again, affinity refers to the drug's tendency to bind to its receptor. If a drug has a high KD, it means it has a high tendency to dissociate from the receptor, KD, dissociation constant, which means you will need a large quantity of the drug to occupy 50% of the receptor binding sites at equilibrium. All right? Drugs with high KD values have low affinity for the receptor. In other words, there is an inverse relationship between KD and affinity. High KD indicates low affinity, okay? On the other hand, if a drug has a low KD, it has a low dissociation constant, implying that it tends to remain bound to the receptor once it binds. It doesn't readily dissociate or separate from its receptor. So drugs with low KD values require a lower concentration to occupy 50% of the receptor sites at equilibrium. All right? Now, if this is still slightly overwhelming, let's subtract complexity again. Let's say you're at a party and you notice that some people are attracted to each other and end up holding hands. This attraction between individuals can be compared to affinity in drug-receptor interactions. Affinity is like the level of attraction between a drug and its receptor, okay? We said KD is the equilibrium dissociation constant, and it tells us the tendency of a drug to dissociate or separate from its receptor, right? Now think of KD as a measure of how likely a couple is to let go of each other's hand at the party. If the KD value is high, it means that the couple has a higher tendency to separate or let go. They're going to let go, all right? And if a drug has a high KD, it indicates that it has a higher tendency to dissociate or separate from its receptor, okay? On the other hand, if the KD value is low, it suggests that the couple is less likely to let go of each other's hand. It'll remain bound. So this is the same way as if a drug has a low KD, it means that it has a lower tendency to dissociate from its receptor. So the affinity between the drug and its receptor can be compared to the attraction between people. While the KD tells us about the tendency of the drug to dissociate or separate from the receptor, just like how likely a couple is to let go of each other's hand at a party. All right? So KD helps us understand the concentration of a drug needed to achieve 50% receptor occupancy, with low KD values requiring a lower drug concentration due to higher affinity. All right, that's affinity. Let's now move on to efficacy. So like we said before, not every drug that binds to a receptor will activate a receptor. Drugs that bind and activate receptors are known as agonist drugs. So here we have an agonist drug binding to its specific site on the receptor, and because it's an agonist, it will activate the receptor, which then initiates a signal transduction pathway within the cell to generate a functional response. All right? So an agonist drug both binds to and activates a receptor. And we also have drugs known as receptor antagonists. These drugs can bind to the receptor, but they don't cause receptor activation. They don't activate the receptor. They can bind, but they can't activate it. An agonist drug, okay, activates the receptor, whereas an antagonist can still bind to the receptor without activating it. And we're going to explore the effects of antagonist drugs later in this lecture. So connecting this with affinity, which is a term we use to describe the tendency of a drug, whether agonist or antagonist, to bind to a receptor, it's an important factor in drug-receptor interactions. The ability of a drug to produce a biological response once it's bound to its receptor is determined by its efficacy. Efficacy indicates how likely a drug is to produce a biological response after binding to the receptor. In other words, efficacy measures the drug's effectiveness in producing the desired effect. Okay, let's connect the concepts we've discussed so far by understanding that receptors can exist in different states. Now, there are various models to describe receptor states, but let's begin with a simple model that considers receptors in either a resting state or an active state. So right here, we have our receptor, denoted as R, in its resting state. When the receptor is in this state, a drug is not occupying it, and it's also not signaling, which means that no biological response is occurring. Let's introduce drug A, our agonist drug for the receptor. When the agonist drug and the receptor bind, they form a complex called AR, which stands for agonist receptor complex. This binding event triggers the receptor to transition into an activated state. So for convenience here, we'll use the notation R*, R star, to represent the activated state of the receptor. Once the drug is bound to the receptor, it's going to trigger a switch that turns the receptor into its active state, leading to a cellular response. Now here's the thing. The affinity of the agonist drug determines the occupation of the receptor or its capacity to bind to the receptor. All right? As we discussed earlier, affinity is measured by the KD, equilibrium dissociation constant, with an inverse relationship between KD and affinity. Remember, drugs with high KD values have low affinity, while drugs with low KD values have a high affinity for the receptor. The efficacy of an agonist drug determines how effectively it can activate the receptor once it's bound. Some agonists have high efficacy, meaning they are highly efficient in activating the receptor, whereas some agonists have lower efficacy, resulting in less potent activation of the receptor. Okay, the key point is that agonist drugs exhibit both affinity, which allows them to bind to the receptor, and efficacy, which enables them to activate the receptor once it's bound. Now, looking at an antagonist drug, let's call it drug B, it can bind to the receptor, but it doesn't activate it. This means that drug B, as an antagonist, forms a drug-receptor complex BR. Okay, and this is influenced by the affinity of drug B for its receptor. So antagonist drugs will still have affinity for the receptors, meaning they can still bind to them. However, antagonist drugs have zero efficacy as they are incapable of activating the receptor once bound. Okay? So it's really important to note these two key pharmacological parameters here. Affinity determines the drug's ability to bind to the receptor, while efficacy refers to the drug's ability to activate the receptor after binding. Okay, both receptor agonists and antagonists exhibit affinity, but only agonist drugs possess efficacy. Antagonist drugs lack efficacy, preventing them from activating the receptor even when they are bound to it. Okay, so receptors can exist in different states, such as resting and an activated state. All right, let's move on. So we've covered affinity and efficacy and things that influence receptor binding and activation, as well as what agonists and antagonists are. Okay, so now let's go through the methods used to quantify and analyze drug-receptor interactions, looking at concentration response curves, or CRCs. Before we do that, we need to first understand, why are we doing this? What's our purpose here? Because there's no point of me going through this, but we're not actually understanding what our purpose here. What's the big picture? CRCs are a fundamental tool for understanding these relationships, the connection between a drug and its functional or biological effects. All right? The concentration response relationship is a fundamental concept in pharmacology, and it plays an important role in understanding the effects of drugs. Okay? And we're going to be focusing on agonist drugs here. Let's break this down. When we administer an agonist drug, the biological response initially increases proportionally with the amount of drug that is given. So in other words, as the drug concentration increases, the effect becomes more pronounced. All right? The increase in drug concentration leads to more drug molecules binding to the receptors through the law of mass action, okay, as we covered earlier. And since agonists have efficacy and can activate the receptor, a higher number of occupied receptors means more receptors transition into their active configurations, into their active state. And what happens then is that there will be an increased activation of downstream signaling pathways and a greater biological effect. Beauty. Initially, as the drug concentration rises, the response increases correspondingly. Okay? Here's the thing though. There is a limit to this relationship, because as the drug concentration continues to increase and reaches the upper range here, the response begins to plateau. This means that even if the drug concentration keeps rising, the response no longer increases further. It plateaus. So we represent this relationship between drug concentration and biological effect using a concentration response curve, CRC. So on the x-axis here, it represents the drug concentration. Okay, CRCs are usually presented on a logarithmic scale. This scale is important because pharmacology often deals with drugs that have a wide range of concentrations. Okay, so the drug concentration is plotted on the x-axis, progressing from left to right with increasing concentration of the agonist drug. The y-axis here represents the response to the drug, which can be a generic term referring to various measured effects, depending on the system that is being measured. But for now, we'll use the general term response. The response increases as you move up the y-axis. Now, when plotting a concentration response curve, you end up with a sigmoidal graph that exhibits a clear bottom plateau, okay, a region of growth here, and a plateau at the top. This sigmoidal shape is commonly observed in most drug-receptor interactions when CRCs are analyzed in this manner. So typically, the linear portion of the curve lies between approximately 20% and 80% of the maximum response. For therapeutically used drugs, this linear region corresponds to the therapeutic range. So within this region, the desired therapeutic effect is observed, all right? And increasing the drug dose within the therapeutic range leads to a greater therapeutic response. The therapeutic range is defined as the target range where the drug concentration is sufficient to produce the desired therapeutic effect without an increased risk of adverse effects, okay? However, when drugs are used at high concentrations beyond their therapeutic range, there is an increased risk of dose-related adverse effects. And for the area below the therapeutic range, this is referred to as this subtherapeutic area. So this occurs when the drug is administered but not in sufficient quantity to achieve the desired therapeutic effect. Okay? For most CRCs, the linear range between approximately 20% and 80% of the maximum response corresponds to the therapeutic range, okay? Now, there are two essential parameters derived from CRCs, which are Emax and EC50. So these two parameters provide insights into drug-receptor relationships and other pharmacological factors such as efficacy and potency. Emax represents the maximum response achievable by the drug under specific conditions, okay? Emax is reached when the drug has elicited the highest response possible. Beyond this point, if we were to add more of the drug, it would not further increase the response, okay? Emax, maximum response. The top plateau of the CRC indicates the Emax value. Again, once the drug has hit its Emax or maximum response, we're not going to produce a greater response if we add more drugs. As the concentration of the agonist increases, with the x-axis progressing from left to right, the response also increases until it reaches the Emax. Okay, so again, even if we were to add, we were to increase the drug concentration by thousands of drug molecules, it would not result in a higher maximum response because the tissue or system has already reached its maximum potential under those conditions. That's Emax, which is the top plateau of the CRC. The other important parameter obtained from CRCs is the EC50, which stands for the effective concentration of 50%. The EC50, this right here, refers to the drug concentration at which 50% of the maximum response to the drug is observed. Now, it's important to understand that the EC50 pertains to concentration indicating where the graph sits on the horizontal axis, okay? In the graph, okay, the maximum response is at 100%. And the line that intersects with the curve of 50% of the maximum response determines the EC50. It indicates the concentration of the drug required to achieve 50% of the maximum response. In this case, the EC50 is right here, approximately 10 to the power of -7 molar. Now, another thing to note is that the EC50 does not equal 50% of the maximum response itself, okay? The EC50 is the concentration of the drug that produces 50% of the maximum response. It represents a concentration value obtained from the x-axis of the graph, not the percentage of the response itself. Okay? All right, let's introduce another concept in pharmacology, which is potency. The potency of a drug is measured by the EC50 value, which represents the drug concentration that produces 50% of the maximum response to that drug. And it provides valuable information about drug activity and the amount of drug required to produce a specific effect. If a drug has a high potency, it means that a lower concentration of the drug is sufficient to achieve the desired effect, whereas a drug with low potency requires a higher concentration to produce the same effect. Let's break this down. Let's say we have 3 agonist drugs, drug A, B, and C. Okay, it's all depicted in the CRC. These drugs have the same Emax, reaching the same maximum response level. The concentration at which the response is 50% of the maximum is marked across the curve, okay, in the middle. This concentration is the EC50 for each drug. In this example, drug A has an EC50 of 2, drug B of 6, and drug C has an EC50 of 30. The drug concentration that produces 50% of the maximum response is what determines these EC50 values. So from this, the EC50 value tells us about drug potency, okay? But what does this mean? How does this actually tell us about potency? The lower the EC50 value, the higher the potency of the drug. Okay? In other words, a highly potent drug with a low EC50 requires a lower drug concentration to achieve the same response. For example, we have drug A here with an EC50 of 2. Okay? It requires a concentration of 2 molar. While drug B with a higher EC50 of 6 requires a concentration of 6 molar. And drug C with an even higher EC50 of 30. Okay? Drugs with lower EC50 values appears on the left side of the CRC and are considered more potent because they require fewer drug molecules or a lower concentration to produce the same effect. Okay? So this is evident when examining the maximum response levels as well. Drug A reaches its maximum effect at a much lower concentration compared to drug B or drug C. Drugs with lower potency require a higher concentration of the drug to produce the same effect. And they have a high EC50 and will appear on the right side of the CRC. Okay? Now, if this is still quite overwhelming, let's subtract complexity again. Okay, this is how we're going to do it. Imagine you have 3 chefs, okay? They're each preparing a dish using a different amount of special seasoning, the EKG Science special, okay? The chefs represent different drugs, and the amount of seasoning they use represents drug potency. Now, our goal here is to achieve a flavorful dish that corresponds to the desired biological effect of the drug, okay? Chef A is a highly potent, okay, and uses only a tiny sprinkle of seasoning to make the dish beautiful, to make it flavorful. Chef B is less potent and requires a moderate amount of seasoning to achieve the same level of flavor. Finally, we have Chef C. It's the least potent and needs a large amount of seasoning to make the dish tasty. Let's now visualize plotting the quantity of seasoning each chef used on the x-axis of a graph and the final flavor of the dish on the y-axis. So this graph resembles a concentration response curve, okay? As you move from left to right on the graph, the amount of seasoning increases. For Chef A, the curve starts at a low seasoning amount, rapidly increasing, and then levels off at a high flavor point. So this indicates that Chef A's dish reaches its maximum flavor, or response, quickly within only a small amount of seasoning or concentration. The EC50 value representing the concentration where the dish achieves 50% of its maximum flavor is at a very low point on the x-axis. Okay? Now looking at Chef B's curves here, it starts with a higher amount of seasoning and it steadily increases in flavor, okay, and levels off at a slightly higher flavor point compared to Chef A. So the EC50 value for Chef B is higher than that for Chef A, meaning a greater response, a greater concentration of seasoning is needed to reach 50% of maximum flavor. Finally, we have Chef C. Chef C's curve starts with a substantial amount of seasoning, gradually increases in flavor, and levels off at the highest flavor point. The EC50 value for Chef C is at the highest among the 3 chefs, indicating that a significantly larger concentration of seasoning is required to achieve 50% of the maximum flavor. Gordon Ramsay is not impressed, okay? So in this analogy, the potency of the chefs represents the potency of drugs, and the seasoning amount correlates with drug concentration. And the CRCs demonstrate how different drugs, in this case chefs, require varying concentration to reach their maximum effect or maximum flavor, with more potent drugs achieving the desired effect at lower concentrations, just as Chef A requires less seasoning to make the dish beautiful. Okay? So the concentration response curve provides us with valuable information about a drug's behavior, including 2 important parameters. We have EC50 and Emax, okay? The EC50, again, indicates drug potency, while the Emax represents drug efficacy. And recall what efficacy means. It refers to the ability of a drug to activate the receptor once it's bound. Now, all agonist drugs have efficacy, meaning they can activate the respective receptors. However, not all agonist drugs have the same efficacy. And this is where we introduce the concept of subdivisions within agonists. Okay, let's slow down for a second. So a full agonist is a drug that can elicit the maximum response in a specific tissue. The top plateau, okay, also known as the Emax, is the highest point on the concentration response curve. So for example, okay, drug A in the graph is a full agonist, producing a maximal response of 100%. Full agonists have high efficacy and can usually achieve the maximum response without needing to occupy all the receptors. Some may only need to occupy a small percentage of receptors to reach the maximum response. That's full agonists. Then we have a partial agonist. Okay, so a partial agonist is also an agonist drug, but with lower efficacy. It can bind to and activate receptors, but has a diminished ability to do so compared to full agonists. In the graph here, drug B and C are partial agonists as their Emax values are lower than 100%. Okay, so you can see here drug B achieves around 80% of the maximum response, while drug C reaches approximately 30%. Okay, so partial agonists can never produce as large a response as full agonists, regardless of the amount of drug added. Okay, so they have limitations in activating receptors, preventing them from reaching the same Emax as full agonists. All right, that's agonist drugs. Let's now move on to antagonism and go through the different types. Okay. Let's remember the difference between agonists and antagonists first. So we said that agonists possess both affinity and efficacy. They bind to and activate the receptor, leading to a biological response. Now, on the other hand, antagonist drugs work by reducing the action of another drug, usually an agonist. So most agonists are receptor antagonists, which is what we'll focus on. Okay? So it still has affinity for the receptor, allowing it to bind to the receptor, but it doesn't activate the receptor or trigger a response, so it reduces the action of agonists. Okay? Let's break this down further. Receptor antagonists can be further categorized into competitive antagonism and non-competitive antagonism. Let's start with competitive antagonism. So right here, we see the agonist drug binding to its active orthosteric site on the receptor, activating signaling mechanisms and producing a biological response. This site, where the primary agonist binds, is known as the orthosteric binding site. In competitive antagonism, both the agonist drug and the antagonist drug bind to the same site on the receptor. On the left here, we have the agonist drug, and on the right, the antagonist drug. They're competing for binding to the same part of the receptor. And due to structural limitations, the agonist and the competitive antagonists can't simultaneously bind to the receptor site. So imagine a crowded parking lot where there's only one available parking space. The agonist drug is like a car trying to park in the spot to activate a receptor. However, when an antagonist drug is present, it also wants to park in the same spot. So just like two cars competing for the same parking space, the agonist and antagonist drugs are competing for the same binding site on the receptor, okay? But only one can occupy the spot at a time, either allowing the agonist to activate the receptor or blocking it from doing so, okay? So this competition for the binding site leads us to an important characteristics of competitive antagonists. All right, as we discussed earlier, drug receptor binding kinetics follows the law of mass action, where a higher concentration of drug increases the likelihood of forming drug receptor complexes. In competitive antagonism, increasing the concentration of the agonist drug can outcompete the antagonist and restore receptor activation. Okay, so if the agonist drug is present in 1,000 times higher concentration than the antagonist, more agonist molecules will bind to the receptor compared to the antagonist, okay? So this ability to overcome antagonism by increasing agonist concentration is referred to as surmountable response. Surmountable means that the agonist effect can be overcome with a sufficient concentration of an agonist drug, all right? You may also hear this referred to as reversible antagonism, okay? So surmountable and reversible antagonism are terms used to describe this type of competitive antagonism. And later on, we'll explore how different types of antagonism can be distinguished using CRCs, all right? So competitive antagonism is the most common type of antagonist for therapeutically used drugs. However, it's also important to be aware of other types of antagonism, such as non-competitive antagonism. In non-competitive antagonism, there are different subtypes, but the key feature is that both the agonist and antagonist drugs can bind to the receptor simultaneously. The receptor and antagonist can coexist and bind to the receptor at the same time. As we discussed before, competitive antagonism can be overcome by increasing the concentration of the agonist drug. So with enough agonist drug bound to the receptors, the maximum response of the agonist can still be restored. This is why it's called surmountable antagonism. Competitive antagonism is surmountable. Now, non-competitive antagonism is a type of insurmountable antagonism. We said that in non-competitive antagonism, the antagonist and agonist drugs can both bind to the receptor simultaneously, but at different binding site. The antagonist doesn't directly compete with the agonist for the same binding site on the receptor. In non-competitive antagonism, the antagonist impairs the function of the receptor or prevents the agonist from producing the maximum, it's a maximal effect, regardless of the concentration of the agonist. This effect is not reversible, by increasing the concentration of the agonist, which is why it's considered insurmountable. So even if you have very high concentrations of the agonist drug, the antagonist will always limit the maximum response. So insurmountable antagonism occurs when the antagonist irreversibly alters the target receptor or its binding site, This irreversible alteration of the receptor can happen through the formation of covalent bonds between the antagonist and the receptor. So this permanent change in the receptor lasts for the lifetime of the receptor protein. Another scenario where insurmountable antagonism can occur is when the agonist drug has an extremely high affinity for the receptor, which means if the agonist has a very high affinity, it remains tightly bound to the receptor and doesn't dissociate easily. As a result, it can be considered effectively irreversible. So insurmountable antagonism can happen either due to irreversible modification of the receptor or when the agonist has exceptionally high receptor affinity, making it difficult for the antagonist to be displaced from the receptor. All right? Okay, we've covered a lot. So far, we have discussed receptor antagonism, where an agonist drug and antagonist drug interact at the same receptor. Now, there's another type of antagonism, and it's called physiological antagonism or functional antagonism. In physiological antagonism, two different drugs have effects that are functionally opposite, reducing the effects or reducing the action of each other due to their inherent impact on biological function. Meaning, they're not antagonizing each other, They are reducing the action of the other drug. In most cases of physiological antagonism, the drugs act through different receptor systems or signaling pathways. To understand this concept better, let's go through an example. So we'll examine the effects of two different drugs on heart rate. We have acetylcholine, which is a neurotransmitter in the autonomic nervous system. It decreases heart rate when it binds to its specific receptors. And on the other hand, we have adrenaline, which is released in the adrenal gland during the fight or flight response, And it increases heart rate by activating different receptors. So the heart contains various receptors, including muscarinic and adrenergic receptors. Acetylcholine activates muscarinic receptors, specifically the M2 receptors, leading to a decrease in heart rate. So acetylcholine is an agonist at the M2 receptors. Now, adrenaline acts as an agonist at beta-1 adrenergic receptors, resulting in an increase in heart rate. So in this example, we can see acetylcholine and adrenaline demonstrating physiological or functional antagonism. Although both drugs are agonists, their functional effects on heart rate are opposite. Acetylcholine decreases heart rate while adrenaline increases it. So we refer to acetylcholine and adrenaline as displaying physiological antagonism in this scenario. All right? So those are the different types of antagonism. Let's now go through how we can analyze antagonism using CRCs. We're just going to be talking about receptor antagonists here, We'll look at both competitive and insurmountable antagonisms and how they will affect the concentration response curves. This is actually pretty cool. So as we covered in competitive antagonism, the agonist drug and the antagonist drug compete for binding at the same receptor site. So they can't both bind simultaneously. So factors like concentration determine which one will be bound to the receptor. This is why competitive antagonism is referred to as surmountable or reversible antagonism. If you have high concentrations of the agonist drug in the system, it can outcompete the competitive antagonists and bind to the receptors. This allows for enough activation of the receptors to achieve a maximum response in the cell, tissue, or system, even in the presence of the competitive antagonist. By increasing the concentration of the agonist, you can overcome the effects of the competitive antagonist. Okay? So this is a characteristic. This is the characteristic feature of competitive antagonism. And we can observe it by examining the effects of the competitive antagonists on the agonist concentration response curve. Competitive antagonism causes a parallel rightward shift in the agonist concentration response curve. This means that higher concentration of the agonists are needed to produce the same response in the presence of the competitive antagonist compared to when it's absent. Okay? Let's draw this out and look at competitive antagonism and its effects on the agonist CRC. Let's start with curve number 1 here. This curve represents the concentration response of an agonist drug. On the x-axis, we have the log scale of the agonist concentration, while the y-axis represents the linear scale of the response. Curve 1 shows that drug A is a full agonist because it reaches the maximum response, achieving 100% of the maximum effect. Okay? The EC50 value of drug A, which indicates the concentration at which 50% of the maximum response is obtained, is represented by the point on the curve where the response is at 50%. This right here. This point corresponds to the concentration of drug A that achieves 50% of the maximum response. Okay? Now let's focus on curves 2 and 3. Which demonstrates the impact of increasing concentrations of a competitive antagonist on the agonist drug. So the purpose of curves 2 and 3 is to show the effect of the agonist drug in the presence of a competitive antagonist. As you can see here, the competitive antagonist shifts the agonist concentration response curve to the right. Curve 2, which represents a low concentration of the competitive antagonist, is shifted slightly to the right compared to the agonist alone. Curve 3 here, curve 3 on the other hand, depicts a high concentration of the competitive antagonist, causing a further rightward shift. Okay, so in the presence of the competitive antagonist, the agonist needs to be in higher concentrations to beat it and turn on enough receptors to get the full effect. Okay? Now, there are 2 key observations to make here. The first one is the presence of the competitive antagonist alters the EC50 values of the agonist drug. It increases the EC50, meaning higher concentrations of the agonist are needed to achieve 50% of the maximum response. All right? The second thing is at high concentrations of the agonist, okay, located on the right side of the graph, it's still possible to reach the full maximum response even in the presence of the competitive antagonist. This parallel rightward shift is what we refer to as parallel shift in the agonist CRC. It indicates that the curve has been shifted to the right without affecting the maximum response. Pretty cool, right? So, to summarize, curve 1 represents the agonist drug alone, while curves 2 and 3 shows the response of the agonist drug in the presence of increasing concentrations of the competitive antagonist. Okay? The competitive antagonist causes a rightward shift in the agonist concentration response curve without altering the maximum response. All right? That's how a competitive, reversible, or surmountable antagonist affects the CRC. In the presence of a competitive antagonist, the CRC shifts to the right, but the maximum response can still be achieved with a sufficient amount of the agonist. Okay? Now let's explore the other type of receptor antagonism we mentioned earlier, insurmountable antagonism, which is sometimes referred to as irreversible antagonism. The key distinction of insurmountable antagonism is that it reduces the maximum response of the agonist drug. Unlike with surmountable antagonism, you can't overcome the effects of an insurmountable antagonist, okay? You can't overcome it simply by increasing the concentration of the agonist drug. This is because most insurmountable antagonists permanently modify the receptor protein or the receptor binding site. Okay, when a drug binds covalently to a binding site, the site is changed in a way that can't be changed back. No matter how many agonist drug molecules are present, they can no longer bind to the changed site because it's been modified. It's been altered. It can't fit in anymore. All right. So then, how does this manifest on a CRC? So let's examine this. On the x-axis, we have the agonist concentration, and the red line here represents the response of the agonist drug alone. And then we have two orange lines. These lines depict the effects of the agonist drug in the presence of an insurmountable antagonist. So here we can see that the presence of the insurmountable antagonist causes as well, a rightward shift, similar to what we observed with competitive antagonism. However, more importantly, we can observe a decrease in the maximum response. In the case of insurmountable antagonism, no matter how much agonist drug is present, when the insurmountable antagonist reaches sufficient concentrations, it prevents the agonist from achieving the maximum response. Okay? So these curves reach a maximum plateau below the response obtained with the agonist drug alone. Okay? To summarize, in insurmountable antagonism, we see a rightward shift in the CRC, similar to competitive antagonism. However, the distinguishing feature is the reduction in the maximum response. Regardless of the concentration of the agonist drug and an insurmountable antagonist, when present at sufficient levels, the maximum response just can't be achieved, okay? No matter how much agonist drugs we add into the system, the maximum response just won't be achieved. Because of this, distinguishing between reversible competitive antagonism and irreversible insurmountable antagonism can be challenging. Because, look at this, at low concentrations, the effects of an insurmountable antagonist can initially resemble those of a competitive antagonist, okay? So, in the graph shown here, the red curve represents the response to the drug. Let's say it's, histamine. And the other three lines show how histamine reacts when an antagonist drug is present in increasingly higher amounts. At a low concentration of 2 nanomoles of antagonist, we can mistake this for acting as a competitive antagonist because it causes a rightward shift without affecting the maximum response. However, as the concentration of the antagonist increases with a higher concentration of the antagonist, we can see a classic insurmountable antagonist response characterized by a rightward shift and a reduction in the maximum response. This shift occurs because the antagonist has irreversibly modified the receptor or receptor binding site. The reason insurmountable antagonists can initially resemble competitive antagonists at low concentration is due to the presence of spare receptors or receptor reserves in tissues. These spare receptors allow the agonist to continue binding and eliciting a response even in the presence of low concentrations of the antagonist. However, as the antagonist concentration increases, it eventually overwhelms the spare receptors, leading to a clear and insurmountable antagonistic effect. Okay? Let's go through receptor reserves. What are spare receptors? So receptor reserves refers to the presence of additional receptors in a tissue or a system that are not required to elicit a maximum response. Okay? So this concept is closely related to the idea of efficacy, which is the ability of a drug to activate the receptor once it's bound. So full agonists, which have high efficacy, can produce a maximum response even when only a fraction of the receptors are occupied, okay? So here, we can see a cell with 8 receptor molecules. 1, 2, 3, 4, 5, 6, 7, 8. You get it. In this example, the maximum response in the tissue is achieved with only 3 out of 8 receptors occupied. This means that less than 100% receptor occupancy is needed for a maximum response. The presence of spare receptors, or receptor reserve, explains why insurmountable antagonists at low concentrations can sometimes resemble competitive antagonists on a CRC. The spare receptors, which are not required for a maximum response, can continue to allow the agonist to elicit a response even in the presence of low concentrations of the antagonist. However, as the antagonist concentration increases, it eventually overcomes the spare receptors, leading to the characteristic effects of insurmountable antagonism. Okay? So, to better understand this concept of spare receptors and how insurmountable antagonist affects the CRC, let's go through an example. So let's say we had a potent full agonist drug with high efficacy. In this case, only 1% of the total receptors need to be bound by the agonist to produce a maximum response in the tissue or cell or system. So due to the full agonist's high efficacy, an irreversible or insurmountable antagonist could potentially occupy the remaining receptors without lowering the maximum response. At low concentrations of the insurmountable antagonist, it initially produces a parallel shift in the CRC. This occurs because, again, the spare receptors or receptor reserve can still allow enough agonist binding to maintain the maximum response. However, as the concentration of the antagonist increases and more receptors become occupied by it, a point is reached where over 99% of the receptors are occupied. So at this stage, the spare receptors are depleted, okay, and it becomes impossible to have enough agonists bound to the remaining receptors to overcome the antagonist effect. This is when the maximum response to the agonist starts to decrease. The presence of the insurmountable antagonist initially causes a shift in the CRC, requiring higher concentrations of the agonist to overcome its actions. But with higher concentrations of the antagonist, there is a clear reduction in the maximum response. Okay, the irreversible antagonist has occupied enough receptors to prevent sufficient agonist binding and activation. Now, it's important to note that the 1% receptor occupancy mentioned in this example, it's not a fixed value, okay? It's not a fixed value for every drug receptor interaction. It simply serves as an illustration of the concept, okay? So the combination of spare receptors, the lack of requirement for full receptor occupancy by some agonists, and the effects of insurmountable antagonists often make low concentrations of insurmountable antagonists resemble competitive antagonism or antagonists. So when this happens, we need more information about the system so that we can draw accurate conclusions. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist.